My name is Marco Sardiello, and I work as assistant professor at the Department of Genetics at Baylor College of Medicine. And I have a lab at the Neurological Research Institute, which is a collaborative effort between the Baylor College of Medicine and the Texas Children's Hospital in Houston. So, my lab is working on a subset of lysosomal storage disorders that are, generally these diseases are inherited diseases that affect the metabolism of the cell and that often result in neurodegeneration. And in particular, we are studying Batten disease, also called neuronal ceroid lipofuscinosis. So, and in particular, one form of Batten disease, which is the juvenile form. The main question is, can we make degradative processes in the cell more efficient? This is a question that is relevant to many diseases. Not only some of the lysosomal storage disorders like Batten disease, but also other more widespread neurodegenerative diseases such as Alzheimer's disease or Parkinson's disease, Huntington disease. All of these neurodegenerative diseases are characterized by the continuous buildup of material within the neurons, or also in some cases outside of the neurons. Because there is, of course, it is coming from some genetic defects or some interaction with environmental toxins. This is the case for some forms of Parkinson's disease. But in any case, all of these diseases have this in common. There is a constant accumulation of material that is not supposed to be accumulated. So this is an aberrant accumulation of material within the cell. And eventually, this will impair the function of the cell. And especially the neurons, the cells in our brain, are very sensitive to this kind of damage. So the general question was, can we make waste management in neurons more effective, more efficient? So a few years back, we discovered that there is a master factor that is called TFEB that basically acts as a switch to enhance the processes that contribute to waste management in a cell. What was missing was a way to act on it, for example, with a drug. So what we have now discovered is that the activity of TFEB is under the control of another protein that is called AKT. And the good news is that there are drugs out there that have been designed to target AKT. So we can use these drugs basically to modulate AKT, and in this way to modulate the activity of TFEB. We can use these drugs to make TFEB more active and more effective at managing the waste material within the cells. So we have tested some of these drugs in cellular and mouse models of juvenile Batten disease, and we have actually verified that activation of TFEB makes waste management in the Batten disease cell more effective, so there is less storage over time, and this will affect neurodegeneration in a positive way. So the study that we have conducted by using an animal model of juvenile Batten disease has shown that the continuous stimulation of TFEB results in a amelioration of the disease. So there is an elongation of lifespan of these mice. There is a reduction in the neurodegeneration, a reduction in neuroinflammation, that are all components that contribute to the disease itself. So the main novelty of the study is that we have identified an actionable pathway to activate TFEB in the cell. In other words, now we know what kind of drugs we can use to make TFEB work for us in patients with Batten disease, and hopefully also in other diseases that, like Batten disease, are characterized by the accumulation of waste material. We hope that this study can be translated into clinical practice. I was mentioning that there are drugs already that are under clinical development to target this particular pathway, AKT, that would act on TFAB. We still need to learn a lot about these pathways and how these can be effective in clinical practice. You know, we are talking of a kind of treatment that should last for the whole life of the patients. And so I'm sure that there is still much to be done and to be understood, but at least, you know, we have opened a door for possible clinical exploitation of the enhancement of the function of TFAB and the enhancement of the clearance pathways in the cell.